I'm an assistant professor at Vanderbilt, and today I'm going to be talking to you about piggyBac transposons to cut and paste DNA. And so we're here in the gene editing, modalities of genome editing session, and we sometimes have these questions about whether cutting and pasting DNA is really editing, because we often think about editing as a single base change. So I would just like to suggest that the answer might be right in front of your face, especially if you're staring at a laptop, because you'll see this edit in the menu bar. And what's there on the edits in your menu bar, if you click on that, you're going to see, okay, you've got cut, copy, and, oh, there it is, cut, copy, and paste. And even undo. Okay, piggyBac can undo. So I'll get to that in a second. So we're going to talk about why transposons, where did piggyBac come from, and how does it work. Some of the features that make piggyBac really unique for genome editing and different from a lot of the systems people are using. And then I'll talk about some of the data that people have generated on using piggyBac for gene therapy, and a little bit on the latest and greatest. There's a lot of power coupling piggyBac with CRISPR-Cas systems. Okay, so I've shown this slide for a number of years, and I don't want it to look like I don't like viral vectors. I do, but there are some advantages of using plasmids. So they cost less, probably an order of magnitude less. There's less likelihood of an immune response. You don't have any risk of creating a self-replicating agent. You don't have existing host immunity that we know of that could be changing due to some of the recent stuff with Cas9, right? You don't have any size limit on the transgene, and you have unlimited promoter choice. But what's the problem with episomal plasmid DNA? After you transfect it, as the cells divide, it's lost. So in this really simplified representation, if you have 3 plasmids per transfected cell, in your next generation you'll have either 2 or you'll have 1. And of course, when this cell divides again, one of those cells won't have a plasmid. And that's a very simple idea. You also have things like silencing that are going to contribute to these plasmids losing their expression. In contrast, integrated DNA is replicated with the genome. So if you change the genome of the mother cell, all of the daughter cells will have that change. Transposons are mobile genetic elements, and these were discovered by Barbara McClintock back in the 1940s and 50s, doing work in maize, looking at corn kernel colors. And a lot of people don't know this, but transposases are the most abundant coding sequence in all of nature. So if you were to take a big hat and fill it with every gene known to man that makes a protein and pull out one thing, chances are you would pick a transposase. And all of us in this room are composed of about 48% transposon-derived DNA sequence. So these things are literally everywhere, all around us. And fortunately for us, not all of that DNA is able to still move, but your DNA is somewhat mobile. So these are mobile genetic elements, and it's now being appreciated that they do move around a little bit, and they can even do horizontal gene transfer. Now that we're able to look at some of the more repeat sequences in the genome, people are finding more and more about transposons and how they're not only in an organism, but even in a sort of ecosystem. Okay, so the one we're going to talk about today is piggyBac. And piggyBac is from the cabbage looper moth. Does anyone in this room garden? Does anyone have a vegetable garden? Okay. Does anyone grow cabbage, broccoli, kale, anything like that? Have you seen those little holes in the leaves on your cabbage plants? That might be caused by this caterpillar. They're really annoying. But fortunately for us, someone took cells and made a cell line out of that moth, and the genomic DNA of that cell line had a sequence in it, and that sequence hopped into a baculovirus. So that's where the name piggyBac comes from, is from the hopping into a baculovirus. And so this was really spearheaded by the Malcolm Fraser lab at Notre Dame. And so they published in the 80s that there were these insertions into the back, and they called it IFP2. And they found it was always going to a TTAA, tetranucleotide sequence. And then in 1996, they published that those insertions also could occur within the genome without the baculovirus infection. They renamed it piggyBac, thank goodness, or we'd be talking about IFP2 right now, and that would get old. And then they defined the minimal IR, inverted repeat and terminal repeat sequences, so the pieces of DNA that you need to get transposition. And they found the catalytic DDD domain. piggyBac is unique because it has 3 of these. It has a DDD. Most transposases have a DDE. And they published in 2008 that the NLS was in the C term, so it has an endogenous NLS. And this is not comprehensive, but I just wanted to point out all the work that he's done on piggyBac. Okay, so how does this work for a gene therapy context? We're all using piggyBac in mammalian cells, probably. So you have 2 plasmids. You have a transposon here, and you have a transposase. And the transposon contains inverted repeat, terminal repeat, short DNA sequences that are flanking a transgene. And that can be anything of interest. That could be a marker gene, selection cassette. It could be your gene therapy, important, missing protein here. And then the transposase is made from this other plasmid, and the transposase actually can bind to the inverted repeat sequences on the ends, and it just cuts out the transposon and pastes it into the genomic DNA. The transposase plasmid gets lost, like I talked about, and then that transposase is degraded over time. So getting into the mechanism a little bit more, since this is an education session, I want you to understand this. So you have TTAA tetranucleotide sequences that are flanking those inverted repeats, okay? And so it's binding those TTAAs, and then there's this like hydroxyl attack that occurs, and what you end up getting is a hairpin. So the transposon TTAA is folding back on itself. And that's unique. So when it joins to the genomic DNA, what you end up with is a hybrid here. So you have in green is the genomic TTAA, and in blue is the transposon TTAA, and you can see each of these ends is a hybrid. Okay, so people have used this to great effect to make cell lines and things like that. This is just showing plasmid DNA transfected into cells with selection, but without any integration. And you can see how many colonies you would get out. With piggyBac, you have many, many more cells that are drug resistant. Okay, so now talking about some of the features. So I'm going to get back to that diagram I showed you because it's really important. So piggyBac is able to do seamless excision. So it can reverse that mechanism I was talking about, and it can cut this back out. And so that blue is going to stay blue, and the green is going to stay green, and you end up with a scarless area here. So people have used that for a lot of different applications. I would say probably the most famous is just making transgene-free iPS cells. So you can transfect in the Yamanaka factors on a transposon, and then create the iPS cells with that integration and kind of longer-term gene expression. And then when you're done with the factors, you can transfect in piggyBac transposase again and cut those out. And so now you have iPS cells without any integration. Okay, so comparing piggyBac to other transposon systems. So first of all, these other ones, they cannot do that trick I was just talking about. So that's a unique feature of piggyBac. But you can see these come from really different families. These transposons are not all the same at all. So Sleeping Beauty is probably the most widely used for gene therapy purposes. It was the first to be used for that purpose. TC Buster and TOL2 are both HAT transposons. They work really well in human cells also. And the assay I'm going to talk to you about today is a colony count assay. Everyone uses this. We just select, or we transfect cells with transposon and transposase, like I showed you. Then the cells get trypsinized, replated in selection media, and only the cells that are resistant to the drug for 2 weeks are able to grow out. And so you can see them very clearly. If you just hold up your plate to the light, you can see colonies formed of clones of one cell that had the transposon integrated. We harvest them, we stain them with methylene blue, and then count those. And so you can see the effect, without transposase you'll only get a few colonies, with you get a lot, and that depends on the dilution that you do. Okay, so my postdoc mentor Matthew Wilson published this in 2007, and it's comparing piggyBac to Sleeping Beauty Tcells. That's a very early generation Sleeping Beauty, but you can see piggyBac had really high colony counts in all these situations of different amounts of transposon DNA. A similar paper was published around the same time comparing piggyBac to Sleeping Beauty 11. It saw a very similar thing. So one of the reasons we think piggyBac transposase might work really well is because it seems to be a little more soluble than some of the other transposases out there. We have a paper in Nucleic Acids Research in 2017 that goes into this in more detail. Another thing I want to show you is the localization. So this is actually mouse liver, and you can see where piggyBac is. It's in pink, and it's on the nuclear periphery, right? And so what's in the nuclear periphery? That's where the open chromatin is. And we also see that reflected in genomic DNA analysis of all the integration sites. So here you have Sleeping Beauty, which doesn't seem to prefer open chromatin. piggyBac does. TC Buster also does. And this is a unique thing with piggyBac. It really likes transcription start sites. So this is the distance to the start site. Here you have right at the start site. And red is piggyBac. And then green is Sleeping Beauty. And blue is TC Buster. So other people have seen this. Many labs have published similar things. piggyBac likes transcription start sites. So if you were going to introduce an oncogene, something like that, I would be careful maybe with piggyBac. But the advantage is you are going to get really high gene expression. Okay, so Nancy Craig's lab found this hyperactive piggyBac. They did a screen in yeast, and they looked at different amino acid changes. And they found 7 amino acid changes that, when combined, could create this hyperactive piggyBac. And so Matt Wilson's lab, they compared different combinations of codon optimization and hyperactivity. And so you can see this very clear result where the HAM7PB, which was mammalian codon optimized, 7 amino acid changed, hyperactive piggyBac was the best, and the insect PB, the original, was the worst. Comparing M7PB to Sleeping Beauty 100X, looking at GFP expression over time in Tcells, it looks like M7PB was very, very good and significantly better. Interesting feature of piggyBac is that because it's integrating between 5 and 30 copies per cell, you can multiplex it. So you can put in multiple transposons at the same time. They're all going to get integrated and expressed. And so here we're looking at different clones. This is a complex where you need all 4 of these genes to be expressed and come together to form a functional complex that generates this signal over here. So we do this all the time with various systems. This is also really useful for inducible gene expression because you can put in a tetracycline, we call it PT-TET-on, tetracycline activator protein, along with your response cassette in a totally different transposon. So it makes it easy to combine different things. You can also put in GFP along with your protein of interest. Okay, so another feature of piggyBac that makes it a little different is that it doesn't require homology-directed repair. It doesn't make a double-strand break, so that can have its uses. The thing is, of course, with piggyBac, it's random integration, essentially. It's just going to a TTAA. So how do you tell piggyBac to go to a specific sequence? It turns out it's really hard. So our lab has made fusions, and here I'm showing a zinc finger piggyBac transposase fusion, a tau piggyBac transposase fusion, and a Cas9 piggyBac transposase fusion. And so with regular piggyBac targeting to the HPRT locus, so we can do this kind of selection, you get 0.1% in that locus. With the zinc finger, it went up to 0.42%. With the tau, it went up to 0.97, so almost 1% targeted. This was my idea. I thought I was so brilliant. Oh, let's put Cas9 with piggyBac, and it's 0%. So it's pretty much protected the locus from piggyBac integration. And other labs have seen similar kinds of things, similar kinds of numbers. And the problem is the piggyBac in the chimera is still super active, and it's still integrating everywhere else. So if you were to just pull out 100 integrations in a cell that got this really pretty good tau2 PB, only one is going to be in the HPRT locus that you wanted to go to. So if anyone has any brilliant ideas about how to make that not happen, let me know. But it still needs some work. It's ripe for the picking. Okay, so let's talk about piggyBac in gene therapy, because it is really active in mammalian systems. So this is just luciferase. This is some older data from Matt's lab. And you can see in the liver, we have much higher long-term gene expression with piggyBac than without. Same thing for secreted alkaline phosphatase. You can also introduce a doxycycline response system, and so they added and took away doxycycline. And you can see this mouse over months is having that response still to the doxycycline. So this is some of my work. We translated hydrodynamic tail vein injection into renal pelvis hydrodynamic injection. And so we were able to get expression of luciferase and other things specifically in the kidney. And we saw a modest effect from adding piggyBac. It's significant, but it's pretty small. But we were able to reverse some of the symptoms in a knockout model of the IGF-1R gene, and where when you do a unilateral ureteral obstruction, you get fibrosis. But if you add back in a piggyBac transposon, we were able to stop that from happening. piggyBac, of course, can be used in Tcells. So this is some of the first work done by Yozo Nakazawa, and he saw that you could put in even a fairly large transposon, so 13 kb transposon. You still have expression after a week. And lately, Richard O'Neill in Matt's lab has started using piggyBac in Tcells for protein delivery. He's put in the erythropoietin hormone and stimulated red blood cell production from those Tcells. And so you can see in anemic mice, they have a normal hematocrit after they're treated with this system. Okay, and I think it's really exciting to combine different systems. So you can combine piggyBac with AAV, and this group found that when they did neonatal injections of the AAV, they got very, very high numbers of integrated transposons. This is a liver, and you can see the difference here with piggyBac versus without. And they were able to, get these mice, which have a ornithine transcarbamylase deficiency, to survive when all of them die at about day 10 to 20 without that treatment. Similarly, people have combined piggyBac with adenovirus and shown that there's a significant effect in the lung of CTFR pigs, or CFTR pigs. So this is showing the correct localization of the CFTR transgene in red, and also a really high % bacterial killing with piggyBac versus without. Okay, so this is a really cool experiment from George Church's lab that I want to show you. They've created a new way to do lineage tracing. So typically, you would use a CRE or a CRE-ER to make a genetic change in a locus in your transgenic animals, and then say, okay, everyone with that change that's expressing TD tomato or LAC-C, they came from that original cell. But I think anyone in this room that's worked with CRE systems knows there's some drawbacks to that. So this is a way to do a similar kind of lineage tracing, but with sequencing. And they were able to put in these homing CRISPRs with piggyBac. So They made 60 integrations in mice, and then were able to see how those homing CRISPR guide RNAs are changing over time. And hopefully eventually, once sequencing sort of catches up with this idea, they would be able to take a single cell from an adult and know exactly where it came from, be able to really map out all of the cells, not only the cells that you're most interested in and are driving with Cree. But I also want to point out, they didn't mention piggyBac anywhere in the paper. So it's like buried in the supplemental method. So a lot of people working with CRISPR-Cas have been using piggyBac, mostly just to select out their clones that they need using drug selection, but they usually don't actually talk about the transposon much in there at all. Finally, there are piggyBac clinical trials, and those are happening right now in the United States. You can look on Poseida's website for more information about that. They're using piggyBac in Tcells to treat various kinds of cancers. There's also trials in Japan and Australia, which I've heard about, but I don't know much about, so I wasn't able to give you a lot of information. But I think it's happening on 3 continents, so that's pretty good. Okay, so let's talk about a summary of piggyBac. So some of the disadvantages: you have random TTAA integration. It's artificially regulated gene expression, so cells that are transfected are probably going to have super endogenous levels of the transgene, and depending on what it is, that could be good or bad. Transfection of plasmid DNA is the primary barrier to getting this into the clinic more. We still have a lot to do to transfect various kinds of organs with plasmids. And you get 5 to 30 transposons per cell. That seems like a lot. So if you're looking for 1 copy per cell, I would recommend looking into Phyc31 integrase. That is a phage integrase from Michelle Kalos' lab, and it integrates 1 copy per cell. You can titrate piggyBac down to 1 copy per cell. That's possible, but I think it's probably better if you're okay with a lot of transposons per cell. Some of the advantages, non-viral vectors, they're easier to make and cheaper to manufacture, right? And you can do multiplex gene delivery, so you can put a lot of different transposons in. There's no size limit. Alan Bradley's group has shown you can integrate a 200 kb bacterial artificial chromosome. So that's very large. So I don't know of a size limit, right? It's highly efficient, and the genes are put in open chromatin, so you get high gene expression. And there's no requirement for HDR. So some sources of piggyBac, if you're interested in using it in your work and getting started, you can look on AdGene. There's academic labs that may give it to you. There's Systems Biosciences sells it. There's other industries selling it. I don't know all the sources. But I want to point this out. I don't work for Vector Builder. I don't know anyone at Vector Builder. But I just want to let you know this is a really easy way to get transposons without having to know much about molecular biology at all. Okay, so you just go to the Vector Builder website. You click on piggyBac, go to design, right? It says from $159 US. And then you can design your expression vector. They have select the number of ORFs. You can do up to 4 open reading frames. Those will be separated by 2 A's. And then you can pick whichever promoter you want. So you can do CMV, CAG, EF1 alpha. They have a lot of others too. You can add in a marker. And so this is all going to go by gateway, I think it's gateway, I'm not sure, cloning into this vector, and then it's already got your inverted repeats. We've been using them lately. It works really well. It takes me, okay, I know what I'm doing, but it takes me about 2 minutes to design something in this, and then it costs the same as an antibody, and they synthesize it in China and ship it to us in a month. Yeah. Okay, and so I haven't had time to talk about every use of piggyBac transposition in mammalian cells, but I would point you to our review from 2015 if you're interested in learning about some of these a little bit more. And I wanted to thank my mentors. Dr. Browning and Dr. Bedford taught me how to clone. Michelle taught me all about non-viral gene therapy, and Matt Wilson, of course, all about piggyBac. And I want to thank my funding sources, and my lab, and all of you for your attention, and the organizers for inviting me. Thank you.
So I have one quick question. You showed the integration mechanism is preceded by hairpin formation at the end of the transgene. So how are they open? Is this an intrinsic activity piggyBac has? Or is this
It's something about, I guess, the catalytic domain is allowing that opening and closing to occur. The catalytic domain is stabilizing that hydroxyl, I think. As far as the actual protein chemistry, I don't really know exactly. But Nancy Craig has done most of that work on the hairpin. And you can find her papers. It's all done ex vivo or like in test tube or in yeast. Yeah.
Okay, thanks.
Hi.
No, I think I have to go to the microphone. Please go to the microphone.
I don't know if the microphone's actually working, but I'll try to speak loud. Okay. I'm interested in the idea of using piggyBac to put large pieces of DNA into mouse oocytes to make transgenics possible, and also what type of size limitations. I mean, are we talking 100, 200 kb?
Yeah, so I would look at Alan Bradley's work. He's done a lot of that. They've integrated, I think it was 220 kb, I could be wrong, directly, with piggyBac. So that shouldn't be a problem if you want to do 100, 200. I wouldn't expect the highest efficiency ever. Yeah.
Okay.
It's only about TTAA. It's very short, right? So many places it will be repeated in human genome itself.
Mm-hmm.
So the moment you overexpress the transposase,
Mm-hmm.
segments in human genome itself will start jumping here and there. Won't it cause a lot of problem?
No,
and you
'cause
told that it can be, the distance between this TTAA can be any length. 2
Yeah.
kb, 3 kb, 5 kb, or 10 kb.
Okay,
Doesn't
so
matter.
the TTAA is the insertion site. The sequence that's required for the transposon to be cut out is much longer than that. It is probably 1 to 200 base pairs is required for the inverted repeat, terminal repeat sequences that are flanking your transgene of interest. So it can hop into a lot of different places in the mammalian genome, but it's not able to cut out a lot of different sites in the genome. So we looked at that. There's a paper from Sunandan Saha is the first author, and it's looking at possible genotoxic effects of piggyBac in human cells. And we predicted some of these that could happen based on homology with the inverted repeats, and we didn't see that hopping happening.
Yeah, thank you.
Yeah.
Okay, I'd like to thank all the speakers for a very original session, and thank you all for your attention, and have fun at the rest of the meeting. Thank you.